Our DNA, the blueprint of life, holds the instructions for building and maintaining our bodies. This genetic code determines our traits, from eye color to susceptibility to certain diseases. However, there is another layer of control beyond the DNA sequence itself, a realm of molecular regulation known as epigenetics. Imagine DNA as a vast library of genes. Epigenetics acts like a librarian, determining which genes are accessible and expressed and which remain silent. During development, cells with identical DNA differentiate into specialized cell types. This transformation is driven by epigenetic mechanisms that activate or silence specific genes. These epigenetic marks can be passed down, ensuring cellular identity. The implications of epigenetics extend far beyond development. Our environment and lifestyle choices can influence our epigenome, impacting health. At the heart of epigenetic regulation lies chromatin, the dynamic complex of DNA and proteins that packages our genetic material within the nucleus. Imagine DNA as a long, thin thread that would stretch for meters if unraveled. Chromatin acts like a sophisticated organizational system, compacting DNA into a manageable size while also controlling access to the underlying genetic information. This intricate packaging is not static, but rather a dynamic and responsive entity, constantly remodeled and reshaped by a multitude of cellular cues. Within chromatin, DNA wraps around protein spools called histones, forming nucleosomes, the fundamental repeating units of chromatin structure. These nucleosomes are further organized into higher-order structures, ultimately determining the accessibility of genes for transcription. The dynamic interplay between DNA and histones is orchestrated by a diverse array of enzymes that modify histone tails protruding from the nucleosome core. These modifications, including acetylation, methylation, and phosphorylation, act like molecular switches, influencing the tightness of DNA packaging and thereby regulating gene expression. The intricate interplay between these modifications creates a complex epigenetic landscape that shapes the cellular response to internal and external stimuli. Neurons, the fundamental units of our nervous system, are highly specialized cells responsible for transmitting information throughout the brain and body. These remarkable cells communicate with each other through electrical and chemical signals, forming intricate networks that underlie our thoughts, emotions, and behaviors. Unlike many other cell types, neurons are post-mitotic, meaning they generally do not divide and replicate once they have differentiated. This longevity poses a unique challenge for neurons. They must constantly adapt and remodel their connections to accommodate new experiences and store memories throughout our lifespan. This remarkable plasticity, the ability of the brain to change and rewire itself, is essential for learning, memory, and cognitive function. While the mechanisms underlying neuronal plasticity are complex and multifaceted, emerging evidence suggests that chromatin dynamics play a crucial role in this process. By regulating gene expression patterns, chromatin modifications can influence the strength and stability of synaptic connections, the junctions between neurons where information is transmitted. This dynamic interplay between chromatin structure and synaptic plasticity provides a molecular framework for understanding how experiences shape our brains and contribute to the formation of long-lasting memories. Section 4. A mouse, a memory, and a molecule. Researchers used mice to study chromatin plasticity in memory formation. They manipulated genes to understand molecular mechanisms of behavior. Focus was on the lateral amygdala, crucial for fear learning. Mice underwent fear conditioning, pairing a tone with a mild shock. This created a fear memory where the tone triggered a fear response. Fear conditioning led to specific histone acetylation in neurons. This suggested chromatin remodeling's role in fear memory formation. Researchers manipulated histone acetylation to observe memory effects. Targeting enzymes, they enhanced or inhibited acetylation in the amygdala. These experiments showed chromatin structure's influence on fear memories. Section 5. Acetylation, fine-tuning the memory machine. Researchers manipulate histone acetylation to understand its role in memory formation. They focus on histone acetyltransferases, HATs, and histone deacetylases, HDACs. Targeting these enzymes in the lateral amygdala affects fear memory formation. Enhancing histone acetylation facilitates fear memory formation. Mice learn and remember the association between tone and shock more quickly. Conversely, reducing histone acetylation impairs fear memory formation. These findings highlight the crucial role of histone acetylation in memory.